Welcome everyone to our renal grant rounds, January 22nd, and it's really my pleasure to introduce our speaker today. Oh, it's Hello?
Here we go. I'm going to turn it up a little bit.
Okay. Well, you see, you already know here.
Try it now.
Okay. What about now? Okay. Super. Well, I was just saying that it's really my pleasure to introduce our speaker for today's renal grant rounds, Dr. Paul DeCaen. Dr. Paul DeCaen is a tenured associate professor here at Feinberg School of Medicine, and he is really an internationally recognized leader in ion channel molecular pharmacology. Dr. DeCaen received his training in structural biology and biophysics at the University of Washington and Harvard University. Before entering academia, he worked as a professional scientist at Pfizer Research and Development for 2-3 years. He gained valuable experience, and now back in academia, he actually has had a very productive career with numerous distinctions, including being a Gottschalk Scholar, Howard Hughes Fellow, and a Fulbright awardee. He has authored many publications in prestigious journals like Nature, PNS, Cell, EMPMO. Now, in his lab within the Department of Pharmacology, Dr. DeCaen employs cutting-edge approaches to investigate the molecular pathology of ion channel-related human diseases, and he has spearheaded the establishment of Feinberg Advanced Cryo-Electron Microscopy and Tomography, FACET core, and this is supported by a strategic initiative from Dean's Office and a block grant from the state of Illinois. This core has been really key in advancing a Northwestern mission to explore molecular mechanisms underlying human disease. And with that, please join me welcoming Dr. DeCaen in our grant rounds.
Thanks a lot. That was a really kind introduction. Really kind. I'll work with the mic a little bit. Okay. It's really great to be here. I typically don't do a lot of work directly with you folks in nephrology, but so I'm really honored by this opportunity to give you an introduction into my lab and our work with ADPKD. And it's a basic science lab. So we are investigating all of the molecular pathologies of specific diseases that cause ion channelopathies, or the diseases caused by dysregulation of ion channels. So our lab has been making strong efforts in interrogating their function with things like electrophysiology or specific sensors, or determining their structures. This is all just a product of really talented individuals in my laboratory. Obviously, I just have the honor of being able to come up here and tell you guys their fantastic results, but it's the efforts of these people who actually bring these discoveries to life. Today I'm just going to focus on one story. We're going to go all the way from molecules to disease phenotypes, and this is the product of two individuals mainly. Eduardo Guadarrama, who's an interdisciplinary graduate student in neuroscience, and Aurea Sarte Palermo, who's done about the lion's share of the work. So Aurea started out as a basic chemist, so doing total synthesis of small molecules at UT Austin, and then came to our lab and decided to pave the way for solving molecular complexes of more complex proteins. So he's gone from the simple molecule to the more richly complex molecules. And his career trajectory is really steep and high. He's starting his own research program here at Northwestern within the year. I'm really excited for him. So nephrology, so ADPKD. So this is a prevalent monogenic disorder characterized by cysts in midlife. It's very common. 11 million people worldwide are affected by this. Because of its midlife onset, there's a tremendous opportunity for developing interventions. These individuals know their history, most of them, and their familial history, so they know that they have a strong likelihood of developing the disease. There is no drug cure. There is a drug that delays a bit of the symptoms. There's a V2R antagonist called tolvaptan. It's been repurposed for ADPKD, but it's important to know that this does not delay really the cyst formation and is in no way a cure, but rather delays by the clinical trials a few years of the renal replacement therapy. There's a clear established genetic component to this disease, which is amazing, right? Because you know which variants are responsible for causing the renal cysts. Those are two genes, which are ion channel subunits called PKD1 and PKD2. Also in their protein form, they're called polycystin 1, polycystin 2. I prefer just to stick with the gene name when I go through the nomenclature. Whether or not I'm talking about protein or gene, it's the same thing. There's a prevailing two-hit hypothesis of ADPKD progression, which has been validated by sequence analysis of the cysts in these individuals. They carry a germline variant in either PKD1 and PKD2, and you manifest cysts later on in life after acquiring a second hit to another allele, another polycystin allele. So you can kind of consider this as either a two-hit or a gene dose type of hypothesis in which these individuals will progress to cystic kidney disease. But despite our strong understanding of the genetic basis of ADPKD, we still don't know how variants alter the polycystin channel function. And so that comes down to the cell biology, the structural biology, and the functionality of these variants and how they affect how this channel functions. And I think that this is the key in order to unlock therapies for what really is this more than 30 years we've known about this monogenic disorder, yet we still don't have a drug cure that directly interacts with the variant proteins to bring about a cure. A large part of the reason why this therapeutic area is still open is because these are pretty tricky targets to work with. And so the polycystins, they're two channel subunits, but they're actually pretty divergent. PKD2 has 6 transmembrane spanning domains. It classically fits within the transient receptor potential ion channel family, like voltage-gated sodium channels and calcium channels. But PKD1 is quite different. It has 11 transmembrane spanning regions. It has an adhesion GPCR-like motif or component in the first grouping of transmembrane segments, which is kind of like a fusion of an adhesion GPCR and a channel-like protein. And these guys then assemble differently. So PKD2 can form homomers, and actually they're voltage-gated and calcium modulated. Whereas PKD1 and PKD2 can also form a heteromer. And we know very little about how these channels function other than to know their biochemical stoichiometry, which is 1 to 3. And it's generally postulated that perhaps cleavage of this ectodomain will actually activate this channel, but this is yet to be demonstrated in native systems. So complex to work with, interesting stoichiometries which can go into homo and heteromeric forms. But primarily, another component of this that also makes it complex to work with is that they localize to interesting membranes within the cell. And so they actually will localize to a primary cilia, which is a hair-like organelle which resides on the apical side of the collecting duct. And so we know that this organelle, for example, has its own resting membrane potential, has its own calcium concentration, and that is largely in part thought to be because of the enrichment of this calcium channel. So we've been making a living out of designing sensors that localize to the primary cilia and, for example, voltage clamping the primary cilia membrane, where we can assess the impact of drugs and disease-causing variants and how they affect the activity or the open events of this ion channel. So here we have the PKD2 channel, which is readily measured from the primary cilia membrane, and each one of these upward deflections represents an open channel. So isn't that really exquisite detail? So I can tell you that in this instance, I have 2 channels open, 2. It's pretty amazing. So beyond that, doing electrophysiology, we've been able to provide some of the first structures of ADPKD using cryo-EM, and of PKD2 using cryo-EM, and this has provided a template for how we can start to process or interrogate how disease-causing variants alter the function of this channel. So this channel subunit, 4 of them, they all come together to form this ion-conducting homotetramer. There's a voltage sensor domain, which is comprised of the first 4 alpha helices. Within that voltage sensor domain are 2 positively charged arginines, which are linked conformational changes which eventually open the gate of the channel, which is set up by the pore-forming domain. And the selectivity filter between pore helix 1 and 2 is a site that we're going to be focusing on today. And I'm going to be talking about 3 disease-causing variants, which are within 9 amino acids from each other. So just by structural biology, you would look at this and say, the outcome of this missense variant is going to be a loss of function, and they're probably all going to be the same. But I'm going to tell you that that's not the case. And this work kind of builds upon where we've already interrogated mutations previously in the top domain of this channel. So these mutations that I told you about, they are located in the pore helix, and it presents an interesting question, not only just so you can try to understand what's the molecular dysregulation of the disease, but it also presents a really interesting biophysical question that has kind of been plaguing a lot of biophysicists in the field. And it's centered upon this controversy that TRP channels either have 1 gate or 2 gates. And the controversy is as such, everyone agrees that the internal gate is a real gate. That is a piece of the channel that iolates and opens in response to membrane depolarization or other modulating moieties. But the controversial component is whether or not this second site, which is located on the external side of the channel, whether it is actually dilating and forming a true gate, or if it's just, operating as kind of a selectivity filter for the incoming ion. So 2 pieces of this work at play, 2 hypotheses to test. So first, we started by just overexpressing the channel and seeing how it localizes to the native environment, the primary cilia, looking for trafficking defects. So we can see for the wild type channel and 2 of the variants, this phenylalanine 629S and this arginine 628C, they localize to the primary cilia with reasonable co-localization. However, there's the cysteine 632R actually is excluded from the primary cilia entirely. So you're already seeing differences. So on one hand, you have a trafficking defect, clear as day. In addition to this, the loss of the cysteine substitution actually causes the cilia themselves actually to become smaller. So they're about half the size. So we wanted to confirm first the trafficking defect, as well as examine what do the channels that do traffic to the primary cilia, what is the impact on the functionality of the channel? So we conducted cilia electrophysiology by voltage clamping the primary cilia membrane. So in this case, we put on an ARL GFP fluorescent reporter, which confirms our ability to visualize so that we can voltage clamp this little membrane. And we carried out these single channel recording experiments where as we stepped to more positive membrane potentials, the open probability of the channels increases. And the voltage, open probability versus voltage relationship follows more or less a Boltzmann type function. And what we observed is for the ones that do traffic, the phenylalanine 629S and the arginine 638C, we have still a Boltzmann type voltage dependent relationship, but the voltage dependence is shifted to the right. And that's an increase in free energy in order to open this channel. So it effectively between 1.5 to 2 times doubles the amount of energy required to open this channel. So that is the mechanism of this loss of function for these two variants. In addition, we confirmed that membranes expressing cysteine 1632R, the ones where the channel does not express to the channel to the cilium, that these patches are empty. There are no single channel events, reinforcing our conclusion that this is a trafficking defect. So we wanted to interrogate. These guys are all close by each other, these variants. Why then does it cause, a difference in trafficking in one case versus another case where the channel has difficulty opening? So we turned to cryo-EM. And so I know some of you probably know quite a bit about this, but cryo-EM has become the mainstay of structural determination. And this is just a graph of the number of GPCR structures have been solved since 2000 to 2022. And we can already see that cryo-EM is overtaking traditional crystallography methods. And so it enables you to, it eases the barrier of entry to solving structures because primarily you don't have to rely on crystallization. Crystallization in X-ray crystallography is a huge barrier. The channels the proteins have to form essentially lattices within space in order to remain happy and unaggregated so that you can resolve those structures. This removes that. And so what happens is you take your protein sample of interest, you freeze it down on some grids, and then you hit it with an electron beam. So just like light microscopy, cryo-ET or TEMs, these transmission electron microscopes shoot a beam of electrons through this gun, and so it's fired at a particular power. So you'll sometimes hear like people who advertise their latest and greatest TEM, they'll say, oh, this is a 100 kV, a 200 kV, or a 300 kV. That just indicates the amount of power that you're driving through. And within this beam, you can, just like a light microscope, it's more familiar to most people, you have a condenser and you have deflectors that more or less shape the particle source or that beam so that it's uniform before it goes through the sample. Cryo-EM used to be something that people thought was kind of laughable in 2005, for example. But major improvements have occurred in the form of detectors, which has transformed the game, and the reason why we're actually able to get at molecular determination. So these improvements came out of the Silicon Valley. The scientists that were local got the winning share and were mostly notified because they were within that community, within these private industries, and they were actually able to solve the first channel structures and molecular complex structures using cryo-EM. So the thing has taken off. And so this is some images of a structure that I resolved. This is a wild type channel. And this is actually some particles where you can actually resolve. You can see the channel itself in different phases and planes that I collected my first time. So with that in mind, we developed FACET here at Northwestern. So most of our peer institutions, Stanford, University of Chicago, they have multiple TEMs. And we've had nothing here at Feinberg for the past, since I've been here. So it's been like a labor of love. Anytime I get a chance, I'm trying to back channel the dean to see what we can do to change that. And 2 years ago, things changed. We were able to, with a strategic initiative, we were able to come up with $11 million to purchase this instrument and its ancillary equipment so that we could bring cryo-EM to Feinberg. So this is just a general idea. This is all below us. This is down into the bedrock of this building. It's 2 floors down. The drywall has just gone up. I'm only here with a blueprint. All our instruments are in boxes or else in my lab. And this is the general layout. So you'll be coming in here if you're interested in this approach, freezing your samples down, protein in a tube at this point. Then you're gonna, and when they're frozen onto grids, you're gonna come over here to the glacios. This is a 200 kV microscope that's been outfitted with some of the best detectors and energy filters that money can buy. If you do a 48-hour data collection with my samples, you can break 3 angstrom easily. We are planning on building cryo-ET capabilities, so this would allow us to visualize into cells and create lamella type samples. And maybe one day we'll get into a 300 kV Krios microscope here at Feinberg. But nonetheless, there is a Krios that has gone into our Weinberg campus in Evanston. So things are really changing around here, and I want to also alert you guys that if you're interested in this, there are opportunities to do this for free and learn it for free. If you can part with 3 weeks, you can go out to any one of these national centers. As long as you have protein in hand, you write a 1-page proposal, you go there, and you can actually come out with reasonable data sets and learn the technique yourself. We at Facet will provide training, but if you really want to jumpstart your intellectual understanding as well as your practical understanding, I'd encourage you to pursue some of these national centers. I participated. It's well worth your time if you're interested. So here, back to the science. I'm done plugging Facet. We wanted to look at what the impact of the pore variants were. So first, like any good biochemist, you have to purify your sample. And what we found is that trafficking defect variant, the one that stuck out as the one that was non-functional, not getting to the primary cilia membrane, this one is completely unfolded. And so we looked at this through fluorescent size exclusion. We also looked at this using a glow melt. And we could see that there are no tetramers, or very little of the tetramers actually being folded, and the protein itself remains as a monomer. So that explains a bit of the trafficking defect and that difference that we see. For the 2 channels that did fold and still were functional in the primary cilia, we resolved their structure at 2.7 to 2.8 angstrom. And what we observed is not major rearrangements in the overall assembly of the channel. Each subunit comes together in a domain-swapped architecture. There are ions in the pore, and nothing grotesquely different. So remember that thing I was telling you about, the external gate, internal gate, what's going on there in more detail? When we look at the phenylalanine substitution, the normal, that phenyl group sticks into a hydrophobic pocket. And when you substitute it with a serine, that polar group still more or less sits in there, but for all purposes, the overall fold of the pore helices and the surrounding pore segments, the channel's still there. It's still more or less retaining everything, right? This is the external side of the channel. And then when we look at the arginine to cysteine mutation, it breaks up this tripartite hydrogen bond network, which you would think would completely descaffold the pore helix 1 from the rest of the channel. And actually what we see is, no, it's still there, stabilized by other things. However, if you go further away from this external site and move into the internal gate, what you find is that there's a collapse of the internal gate, which is allosterically driven by mutations on the external side of the channel. And so to us, this collapse and it can be highlighted by this disruption of hydrogen bonds that are formed here, and they're forming a bit asymmetrical. You can see how this is pretty much impossible for a partially hydrated calcium or sodium ion to squeeze past in this little place. This mutation fits really nicely with our functional data, in which the mutation stabilizes a closed state of the channel. So this fits very well. And when you think back to the one-gate, two-gate hypothesis, what does it tell you? It tells us a bit that really that external gate probably isn't opening at all or doing much of the gating function at all. And many of the previous studies that have been done in functional work from other TRP channels like TRPV and David Julius's work, where they see effects when you mutate out that external sites, it's probably all driving allostery down at the base of the channel. So using super resolutions, super resolution, so this is from the trafficking data, the cilia electrophysiology and the cryo-EM approaches, we see that there are mechanistic differences with these three variants that are within nine amino acids from each other. So they either alter the channel's function in a loss of function effect, or else they disrupt the ability of the channel to assemble and then traffic to the primary cilia. And the unexpected findings have some significant implications. It really shows that you really have to approach ADPKD perhaps a bit particular to the type of mutation. So on one hand, you can approach this with a corrector, something that Vertex is very much involved in and has done well for CTFR, a chloride channel, to bind and stabilize and correct that energy minima that these channels are falling into and misfolding. Or you have to approach the channel as a modifying approach, where you bind and enhance the open probability of the channel, which is how so many ion channel drugs work. And of course, the allosteric regulation that sites on the external side of the channel can have big effects more than 20 angstroms away at the lower gate of the channel. So we've gone through a lot of the in vitro, the molecular. What is, the mechanistic differences? Do they really actually drive differences in cystic propensity in vivo? So what we did is we initially just made gene variant substitutions within the genomes of mice. So we used CRISPR-Cas9, and what we've done in this case is we have heteromers, which more or less develop pretty normally, right? These have sporadic cysts. It's not very penetrant in these animals' phenotype. However, homomers, actually, they're lethal, and they don't make it post-birth. So between E14 and 17, you lose most of the litter. And if you just study the animals that make it out of their dams, then you end up with only heteromers and wild type. So when we examine their developing hearts, we find that the homomers of either variant actually have septal defects and other cardiovascular abnormalities. You can already see a lot of hemorrhaging that is happening in these embryos. And when you examine the primary cilia of their kidneys, they are blunted. They're much shorter. And in case of the misfolding variant, the homozygous, from the homozygous mice, you can see that there's pretty much no cilia here. So, right. So this doesn't really tell us much about the disease phenotype, right? Because they're dying in utero. And we know that from the two-hit hypothesis that individuals with PKD only have two hits mid-life. They're not born with two hits in their germline. So we set about actually trying to developing a mouse model that recapitulates the human condition. So what we've done is we've created a flox allele. We've crossed it with our disease-causing variants. And so each one of these animals have a functional one functional PKD2 allele until we expose them to doxycycline in their drinking water. And the study carried out like this. So we put about 4 molar doxy in their drinking water for 2 weeks from 1 to 2 month old mice. So these are kind of older mice. They're no longer with their mums. And then we MRI imaged at every half month for 3.5 months. So for the folding variant, several of the mice didn't make it through the study. They had to be culled because they developed such profuse cysts that it was just too much animal for suffering for our protocols. The rest all developed cysts and that rapidly progressed through that 3-month duration. So this is a really powerful way to kind of recapitulate the human disease phenotype, where you can delete one allele, much like the genetic insult that ADPKD patients receive through their life. And so in addition to this, we looked at their cysts, their kidneys post studies, and we can obviously see beyond the MRI analysis that these kidneys are pretty much 3 times as large. It's no wonder you can see that these animals are very sick through this short time period. So this also presents a great challenge because now we have a way to test drugs in vivo and do it in a very clear fashion. So up to this point, there hasn't been, other than homozygous PKD1 variants, where the animals go cystic. And in this case, the cystic phenotype is much more aggressive, and it creates a really nice shorter-term model in order to assess therapeutics or experimental therapeutics. And so I should also add, this is the first time this has been done for PKD2 period. So we've gone from molecules to cystogenic propensity. When you look at the misfolding variant, right, you actually see that this one, which is excluded from the primary cilia, has a more aggressive phenotype, more rapidly forming cysts, which fits very nicely with our in vitro data, where we could not, for example, even get their structures because these proteins misfold so badly. So once again, I want to emphasize the work of Eduardo and Ori, a student and a postdoc, soon to be faculty. I want to thank my funding sources. Thank President Biden for his last 2 days in office. We got awarded the Early Career Award. Who knows what the next months will bring. And thank you for listening to me. I'm happy to address any questions you have.